In this video, we're going to look at some of the research that has led us to our understanding about the structure of DNA, what it looks like, and how it works. So we know that DNA is a nucleic acid. We know nucleic acids are long, slightly acidic molecules built from monomers called nucleotides. So this is what a typical nucleotide looks like. DNA is made up of these nucleotides joined into long strands or chains by covalent bonds. In the case of DNA, it's a specific type of bond called a phosphodiester bond. Nucleic acids are made up of these building blocks called nucleotides. So nucleotides have three parts. There's a 5-carbon sugar called deoxyribose for DNA, a phosphate group, and a nitrogenous base. And there's 4 of those. So here's our nitrogenous base. Here's our 5-carbon sugar. And here's our phosphate group. So here's the 4 kinds of nitrogen bases: adenine, guanine, cytosine, and thymine. And it's the arrangement of these bases that makes up our genetic code and the genetic code of all living things. So here's one nucleotide joined to several others here. There's the covalent bond, the phosphodiester bond makes up the backbone. So Erwin Chargaff, a Russian researcher, was studying DNA, and he noticed that in all organisms, the ratio of A and T, the amount of A's and the amount of T's were the same, and the amount of C's and the amount of G's were the same. Now, it wasn't always, the same from organism to organism, but within that organism, A was equal to T and C was equal to G. He didn't really know what it meant. Later on, Watson Crick figured it out. Rosalind Franklin, worked with Maurice Wilkins and did X-ray crystallography or X-ray diffraction studies. And she used this to show that DNA is a helix. And you can see the X-ray pattern for DNA on the right of Franklin's picture. And it also showed that there were likely 2 strands that made up the molecule. And the nitrogen bases were near the center of the molecule. So Watson and Crick come along and they take their own research along with researchers like Chargaff and Franklin, and they come up with a model that shows the structure of DNA in very precise detail. And it shows that DNA is a double helix in which the 2 strands of nucleotide sequences are wound around each other, and there's very specific things in their model about the spacing and how far apart the strands are and so on. So in Watson and Crick's double helix DNA, the strands are anti-parallel. That means that they run in opposite directions, and that allows for complementary base pairing, where the A's are always paired up with T and the G's are always paired up with a C. So we know already that the sides of the DNA molecule are held together by these special types of covalent bonds called phosphodiester bonds. But the center, the bases, are held together by hydrogen bonds. And hydrogen bonds are much weaker. They're easier to break, and that's going to be important later on. We also know that cytosine and guanine are held by 3 hydrogen bonds, whereas adenine and thymine are held by 2 bonds. We also notice the shape. Some of the bases have a double ring, some have a single ring. The double rings are called purines, and the single rings are called pyrimidines, and you always have a purine and a pyrimidine together, and that keeps the spacing equal. So again, you have A's and T's, C's and G's go together.